Bismillahirrahmanirrahim. So our next video about the Bohr shift. What is Bohr shift now? Bohr's shift, or we can say Bohr effect. Bohr effect. In the previous video, I told you about the partial pressure of oxygen and percent saturation of hemoglobin. Once again, the same graph is in front of you. We increasing the partial pressure of oxygen and it affect the percent saturation of hemoglobin. You can see here two dissociation curve. One has been shown with red and one has been shown with black. And the partial pressure of oxygen remains constant for each curve in the same situation. So this black curve responding to different partial pressure of oxygen at fixed carbon dioxide, and the same in the red curve, fixed carbon dioxide. But the carbon dioxide partial pressure in the red is higher than the black one. That's mentioned here, less partial pressure of carbon dioxide and more partial pressure of carbon dioxide. So what does Bohr says? It says that by increasing the partial pressure of carbon dioxide, the dissociation curve is shifting toward the right, like here. When we increase the carbon dioxide concentration, partial pressure, the curve is shifting toward the right. The black one is on the left and the red one is toward the right. So whenever a curve is shifting toward the right, it means that the percent saturation has been decreased. Like let's see, at partial pressure of oxygen 6, it's maybe like at this point. So at partial pressure 6, if you see the red line here, so it basically shows the percent saturation of about 34% something. So like it says like maybe here, and that's 34%, the percent saturation at partial pressure of oxygen 6, when the carbon dioxide concentration is higher in red. But in the same partial pressure of oxygen, if we join this line like here, so you can see that the partial pressure has, now the percent saturation here is somewhat greater than this one. It's near about 57% at the same partial pressure. Although the partial pressure of oxygen remains the same, but higher partial pressure of carbon dioxide decreases the saturation. Why? It can be explained on the Bohr shift mechanism effect. Shifting of the curve toward the right means lesser saturation if carbon dioxide increases. What are the factors behind this? In the previous video, I told you that whenever the carbon dioxide partial pressure increases, the percent saturation decreases and dissociation will be increased. So yes, here should be more dissociation in the red curve. Hemoglobin more readily unload the oxygen if there's more carbon dioxide. Let's see the mechanism. What is the mechanism basically? What is the mechanism of Bohr shift? Right, the mechanism. Look, here is the blood capillary. This is the blood capillary. This is the wall of the blood capillary, and this is the lumen of the capillary. So the lumen of the capillary. Here we have the red blood cell, RBC. Here is the RBC. Here's the RBC. So here we have plasma, blood plasma, and this is the lumen, and this is the RBC. Here is the RBC. Now in the surrounding area we have cells, of course, because it's a respiring tissues, so there are many cells which are under the state of respiration. Here is the respiration, and of course here should be the tissue fluid. This blue spots, the green spots represents the tissue fluid. So here is the tissue fluid. Here is the tissue fluid. Now, since there is respiration, the state of respiration is taking place here, and in respiration, carbon dioxide is going to be produced in the cells. So this carbon dioxide will be initially dissolved here, passed into the tissue fluid. Here is the carbon dioxide from anywhere, carbon dioxide in the tissue fluid, and this carbon dioxide passed through here in the blood capillary, and in the blood capillary this carbon dioxide pass into the RBC. Now in the red blood cell, we are now in the red blood cell. Here is the red blood cell. Here is the red blood cell. Here is the RBC. Now this carbon dioxide react with the water in the cytoplasm. Water in the cytoplasm of the RBC cell, because it's a cell, so it must have cytoplasm, and it is a reversible reaction. Look, here's some short space, so I'm going to draw this equation here. Carbon dioxide plus water, a reversible reaction, and it makes carbonic acid, that is H2CO3. But this is a reversible reaction. Now here at this step there's a question in the mind. Do you think we don't have water here in the tissue fluid? We don't have water in the plasma? Yes, we do. We have water is even in the plasma and in the plasma and in the tissue fluid. Then why carbon dioxide do not react with water here and here? Why here? Water is placed in the tissue fluid as well as in the plasma, but it doesn't react here, but it reacts in the RBC. Why? The answer is that because in RBC we have an enzyme, and that enzyme is responsible to catalyze this reaction. In the absence of this reaction might not take place. That's why it's not taking place in the tissue fluid and in the plasma. And this enzyme is called carbonic anhydrase. Carbonic anhydrase. Keep in mind the reaction is taking place here in the RBC. So this reaction is taking place in the RBC. This reaction is taking place in the RBC. Now what is the product here? It's H2CO3. But this reaction is like somewhat not so much stable and not remain for a longer period of time, and it immediately dissociates. This reaction immediately dissociates. This H2CO3 is broken down once again. It is decomposed, ionized immediately into H plus ion and plus carbonate ion, hydrogen carbonate ion. Now in this reaction you can see here H plus ion and HCO3 ion, because H2CO3 has been decomposed into its respective ions. Now hydrogen ions, there's a chance that these hydrogen ions might remain in the plasma because they can pass the channels from the RBC, and if it is in the plasma it can make the blood more acidic, because hydrogen ion is responsible to cause low pH. And this hydrogen carbonate ion may change the charge potential of the cytoplasm, because if the hydrogen ion, like this reaction takes place in the plasma actually. This reaction is taking place in the plasma here. So once the H2CO3 has been formed, it can pass out into the plasma from the RBC where it dissociates into H plus ion and HCO3. Now this hydrogen ions should not remain for a longer period of time in the plasma, otherwise it will lower the pH and the blood will become acidic. So this hydrogen ions pass to the membrane of the RBC by facilitated diffusion, and here it reacts with the hemoglobin. Hb for hemoglobin, and hydrogen ion will react with the hemoglobin. Now keep in mind that this hemoglobin is not in the form of hemoglobin, but it's carrying oxygen from the lungs. So it may be in the form of HbO2, because it is usually oxyhemoglobin, and hydrogen ion is going to react with the HbO2. Now here is a displacement reaction because hydrogen is more active. I mean, not in the sense of active, I'm talking about the chemistry. Basically, Hb has more affinity with hydrogen ion as compared to oxygen. So if there is hydrogen ion, there's hemoglobin more likely to accept hydrogen instead of oxygen. So this reaction is also, reversible. And what is going to occur? Here H and this H react with the hemoglobin and it makes HHB, and this is called hemoglobinic acid. Hemoglobinic, which is a compound, hemoglobinic acid, and it will release oxygen. Now this oxygen can easily diffuse out from the RBC because now it's unloaded by this hydrogen. Now there is a problem, one more problem here, because the H2CO3, HCO3, which has a negative charge, if it is also in the plasma, then there's a problem that there will be a charge difference because the blood plasma will become more negative because H plus ion have been removed by the hemoglobin, and the only radical in the plasma is HCO3. Now this HCO3 cannot come back into the RBC like hydrogen. So what should we do? Instead of HCO3,